Hello everyone. In this video, we are going to discuss about the calorimeters and types of calorimeters. So, what is calorimeter? Calorimeter, as the name indicates, it is going to measure the color substance of a fluid or a solution. So, color concentration could be obtained by passing a color light intensity, by passing a light from a lamp or some any source of energy, by passing a color, by passing a light through a substance, the color concentration of that particular substance could be noted. This is known as to be colorimeter. So usually the optical color filters has been used, such as LED or laser or any photodiodes could be used to measure the colorimeter. So here usually the laser LED has been preferred since it is going to be a monochromatic of color. So calorimeter. So calorimeter, here the calorimeter is a experimental setup of this calorimeter. So here the curvet or a beaker, here we'll have a two beakers or the curvet where it is going to be a two beakers have been placed. So the beaker is going to be filled up with the concentration solution, where the concentration of the solution is taken as to be the C, and the length of this beaker is measured as to be the L. So, as I have told you, the calorimeter it is used to measure the color concentration of the substance with the help of an optical light density. So, here the optical light from the lamp is allowed to pass through a filter. The filter filters out some of the basic colors and passes only a colors which has an intensity of I naught, and this curvet or the beaker receives an I naught as the light intensity, and a few of that concentration will be absorbed by the substances, and part of it will be leaving out of the curvet or the beaker, which is going to be the I1. So this I1 will be having a less light density when compared with that of the I naught. When this I1 is allowed to pass through the curvet or the beaker, so it is going to get the I2 will be the outlet of this beaker, whereas the I2 will be the lowest of all the light intensity, where I1 will be lesser than that of the I0, and I2 will be the least of all the cases. So the absorption of this calorimeter is given by the formula A equal to A Cl, where C is the concentration of the absorbing substance, L is a light path with the length. We have taken the length of the curvet, which has to be L, so that L is called as to be the light path of the curvet. Where C is going to be the concentration where it has been absorbed, and A is going to be the absorbability. So now next move on with the second type of the calorimeter, which is the filter photometer. This is also a type of calorimeter where here all the calorimeters could the color substance could be measured with the help of an optical light intensity. So here the lamp is there. The lamp filters out a many the from the lamp many scattered wavelengths of light has been obtained, and that wavelength of light will be filtered out, and particular range of the wavelength is allowed to pass through the C reference and CES, which is going to be the sample cells and reference cells. When it is passed through a sample cells, I mean your reference cells, I mean the sample cells, whenever the sampler is not placed, whatever the output we are getting or we are obtaining at the potentiometer will be same as that of the amount of light which has been passed from the filter. Whenever a sampler is placed before the sample cells, that is the yes is a sampler whenever it has been placed, so the C reference, the output of the C reference is going to get splitted into two halves. One is going to be for the sample cells and the other one will be for the potentiometer. The galvanometer works until there is a deflection in the potentiometer. So it is a type of filter photometer where it does not, why the name comes back for the filter photometer is, so it does not pass through all the lights from the lamp. It just sends the particular wavelength of light which could be acceptable by the filter, where the filter is designed in such a way that it could be acceptable for all the cases. And the third type of calorimeter we are going to discuss is nothing but the sodium potassium analyzer, or which is nothing but the flame photometer. So next is the flame photometer. And this is a flame photometer where we'll have a beaker. Inside the beaker there will be a solution of substances, and the lithium and sodium potassium ions, a few of lithium and sodium potassium ions are also mixed up inside this sampler solution, and it will be burnt in form of flames. From the flames it is being passed through a filter, and from the filter not all the colors have been scattered out, it is get diffracted and through the filter, and from the filter the red, yellow and violet beam has been passed through the photo detector. The red is for sodium, yellow is for sorry, red is for yeah, red is for your lithium, yellow is for sodium, red is for lithium, yellow is for sodium, and violet is for the potassium. So all the lithium, sodium, and potassium ions have been reflected by the photodetector, where we'll have a diodes in the form of photodetector. The light beams are absorbed by this photodetector, and we'll have a potential divider followed by the photodetector, where it just passes out only the sodium ions and the potassium ions at the output of the sodium potassium analyzer. So how the sodium potassium analyzer works is, depending upon the light rays which has been diffracted from the filter, red is for lithium, yellow is for sodium, and violet is for potassium. It is get absorbed by the photodetector, followed by the potential divider, where we will be getting back the sodium and potassium at the output of the op amps. So in this video we have discussed about the calorimeter and types of calorimeter, filter calorimeter, and a flame photometer, which is nothing but the sodium potassium analyzer. Thank you all.